Hello everyone, my name is Susan. Welcome to today's webinar. We are happy to have Dr. Victor Gorobovskaya as our guest speaker. Victor is the Senior Director of R&D and Business Development from ProMed. Today's topic is lipid nanoparticle synthesis and applications. So let's start. So first, what is the lipid nanoparticles? Lipid nanoparticles are self-assembling structure of natural or synthetic lipids in aqueous environments. So the lipids will have hydrophilic head groups and hydrophobic tails. When they dispense in water, they will self-assemble together to form a lipid bilayer structure. Under the help of the cationic or ionizable lipids, nucleic acids such as DNA or mRNA can be packed inside the aqueous interior of the structure. So this is illustration of the self-assembling process under microfluidic mixing methods. So since the success of COVID-19 mRNA vaccines, more and more therapeutic and vaccines based on mRNA lipid nanoparticles have been developed. The table here is summarized the recent clinical trials of mRNA lipid nanoparticles vaccines against the infectious disease and cancers, such as COVID and also rabies virus or melanoma or HPV cancers. Besides the mRNA lipid vaccines, T-cell engineering is another hot topic about the application of lipid nanoparticles. Compared to electroporation, mRNA lipid nanoparticles shows higher cell viability and shorter treatment time. Recently, Ulrich and their colleagues demonstrate an in vivo therapeutic approach to generate transient antifibrotic T-cell CAR-T cells to treat cardiac injury. The next question is how to prepare lipid nanoparticles. Conventional methods include the film rehydration, solvent injection. However, this kind of conventional methods usually generate particles with large PDI and poor reproducibility. Although they can use the extrusion to improve the homogeneity, however, it needs extra steps, resource, and is very difficult to scale up. On the other hand, the microfluidic mixing method can provide a reliable and convenient way to prepare lipid nanoparticles. The schematic here actually shows you that the mechanism of this mixing method. Two steams, one is the aqueous phase with DNA, mRNA, or protein, will mix with the lipids in envelope in a thin microfluidic mixing channel. By controlling the flow rate and mixing time, we can control the size of the lipid nanoparticles in a homogeneous and convenient way. PC Genome provides a fast and reliable solution for the lipid nanoparticle synthesis, which is called a nanogenerator system. Nanogenerator system has a full production line covering from the discovery, development up to commercial scale-up solutions. Next, let's have a look at the performance of the nanogenerator FastM system. First, users can easily control the particle size by changing the parameters, such as the total flow rate or the flow rate ratio. Here, the data shows using the same formulation by changing the flow rate from 1 milliliter per minute to 3 milliliter per minute. You can change the size from around 70 nanometer to 50 nanometers. Even doing so, the size is still keep the homogeneity during changing a parameter. You can see that the PDI, referring to the polydispersity index, will still remain below 0.2 for all of the samples, which indicated that all the samples had a very good homogeneity in the solution form. Another feature of the mixing method is the reproducibility or the batch-to-batch consistency. You can see that here, even generate with different models or different batches, they can still keep a very similar size and similar PDI. And this feature is extremely important during the scale-up production. The next slide, this slide will showing you that we're using a DNA as a payload sample to encapsulate in a lipid nanoparticle. We use a GIP reporter gene as a sample. The DNA will nicely capture it in the LNP with the size around 80 nanometers and with a very nice encapsulation efficiency. You can see that up to 90% of the DNA have been encapsulated in the LNP. And then the next will be a result of the transfections. So we use this DNA-LNP to transfect human cell lines such as K562 or HERG2. In either cell lines, you can see that the cells can be nicely transfected and express the GFP after 48 hours. So next, welcome Victor to share her experience to using the microfluidic mixing method to synthesize the lipid nanoparticles and what's the applications. Hi, Victor.
Yes. So, hello everybody. Recently we bought two machines, nanogenerators, Flex S and Flex M. And this allows us to encapsulate mRNA to nanoparticles. So, Pramap Biotechnologies is generating monoclonal antibodies and doing hybridoma sequencing and also human antibody screening, antibody humanization, bispecific antibody engineering, and stable cell line generation and antibody production. And also recently, we started using our antibodies for generating CAR-T and CAR-NK cells. And we do also different CAR discovery from antibody to CAR generation and in vivo models. And then also we have GMP facilities and develop CAR-T cells for clinical research. So, and on the next slide, I will show you how mRNA can be used with nanogenerator and creating nanoparticles. So we can encapsulate both antibodies and CAR structures using LNP mix and encapsulate with nanogenerator and use it for immune cell therapy and expansion of immune cells, dendritic cells, T cells, gamma T cells, B cells, NK cells, and macrophage cells can be used with nanoparticles to generate. So we first use DNA template on number one. We have DNA template that has coding region of the gene. It has 5' UTR and 3' UTR and poly A tail. On the second, we see that this DNA template with T7 promoter in vitro transcription reaction generate RNA with 5' UTR, 3' UTR, and CAP1 on 5'. So then we test this RNA number 3 on agarose gel, and you can see that we have very good bands of RNA. And this RNA is very stable because it has CAP1 structure. It has very high translatability, stability, and has minimal immune response because we use DUTP and DCTP modified nucleotides. So capping also product generate very good capped RNA. And then we encapsulate with nanogenerator with LNP mix and generate, for example, LNP containing CAR mRNA. So on the next slide, you can show how we used RNA GFP. So on previous slides, you saw DNA GFP, and in this case, we use RNA GFP, and you can see that after transfection to 93 cells, you have more than 97% positive cells with EGFP, RNA, LNP. So it's very effective delivery of RNA, LNP, and this nanogenerator allows you to create very stable expression, and up to 72 hours. We tested even 144 hours, and this RNA still had very high expression of GFP. So on the next slide, you can show we used a spike protein from COVID-19, and we can also this big RNA, more than 4 kb RNA can be generated, and S protein and also Omicron S protein are generated up to 41% positive 293 cells. This was done by FACS with after transfection of RNA and spike protein. You can see that 41% cells of 293 were positive with S. So you can not only you can produce S big proteins with this RNA technology. And on the next slide, on the previous slide, sorry, it was electroporation, but here, yes, electroporation. And on the next slide, it was done with LNP technology. So it's even produced more than 40%, 51% positive cells after 48 hours, which is better than electroporation, and cells are more viable than electroporation. So using RNA-LNP is much better than electroporation. And we can also produce CAR proteins like this. You can show next slide. So Pramab Biotechnologies using nanogenerator created several products that if you can buy this machine, nanogenerator, and use these products already pre-made RNA-LNP, and you can test how use it as a controls. We have GFP, we have spike protein, and RBD protein, and CD19 CAR, BCMA CAR, and HER2 CAR, and also bispecific antibody, EpCAM CD3, and all of them were tested and with quality control and expression in 293 cells, and in terms of CAR cells and NT cells after transfection, and we had more than 50% expression, up to 90% with CAR-T cells, generated CAR-T positive cells. So in conclusion, this technology, nanogenerator and in vitro RNA transcription can be used for successful expression, transient expression of genes, and it has much more advantages than electroporation, and it does not require viruses like lentiviruses that are inserted, and it can be very used as in your research. It is very fast and efficient.
Wow, thank you, Victor, for your sharing your experience. It's very incredible, especially the transfection rate is so incredible. It can up to 90% of the cells, for example, the control one. So is any special, how to say, kind of treatment to how to achieve the 90%? So do we need to pre-treat the T cells, for example, before we transfection, or we can just run right after the fresh separation T cell can also achieve this incredible transfection efficiency?
Usually we take PBMC and we isolate T cells and we activate with CD3, CD28 beads and magnetic beads. And then after 10, 12 days, they are highly activated, they are very highly expanded. Then we use CAR RNA and in nanoparticles, and we add this transfer these cells, and the same with other like NK cells. So you need to activate and expand, and this is very good for manufacturing for example, allogeneic therapy. So you can manufacture from healthy patients, and then you can add of highly expanded, because patients that are sick, the T cells can be less expanded, but in this case you can use from healthy donor and have your modified CAR. Yeah.
Okay, it's very nice. And also I'm very curious on previous slides, then you're showing that you can incorporate some antibody on the surface of the the nanoparticles. So let me see whether it's this slide. So is it this modification is post modification, or you are doing it just during the synthesis? So is any special, is it for a targeting purpose for this incorporate the antibodies on the surface?
No, this would be secreted antibodies, for example, with FC,
Mm,
and
FC.
you encapsulate with nanoparticles, and then you transfect to cells, and for example, to 93 cells, and they will secrete this proteins, and you will use it. You can purify these proteins and use in your functional analysis. So it's very convenient for research to use it, and in future we are planning to do it as a production in vivo. Yeah.
Oh, okay, I see. Okay, it's very smart strategy. So yeah, I think it's a very nice presentation, and I'm just such amazing that Pullman can starting from the synthesis of the gene material up to the end product, the LNP delivered to the cells or even the animal models is very nice.
Yes, we did already in mice injected proteins, and we created antibodies against spike protein. So these nanoparticles are very good used in vivo, and we also did toxicology study. I did not show slides, but there were no toxicity with nanoparticles generated. And also we have also machines, systems that can measure size of nanoparticles. It's like 70 to 100 nanometers, and encapsulation with ribogreen assay. So this system nano generator helped us in research to generate this CAR-T cells, and antibodies and proteins can be used for vaccines and many applications. Yeah.
Okay, thank you. So one last question. So how to use content is about the relationship between the size of PDI in terms of the transfection efficiency. Is some correlation between them? Is any specific size will be usually give a better result?
We are studying this still. There is no correlation between the size of nanoparticles, and mostly it depends on the sequence of your gene and the complex but if nanoparticles too big, yeah, then it's not efficient. Yeah.
Okay, so more related to the gene
yes.
itself, how it can be expressed, but the particle size is not very critical in the cell transfection.
Yeah, we have found that there is variations between 80, 100. There is no correlation. It's a whole complex of the gene plus nanoparticles. Yeah.
I see. Thank you. So thank you, Victor, today to share her experience. It's very incredible and informative. So let's have a summary to wrap up today's webinar. We're happy to introduce all you guys to our nanogenerator FRESH-S and FRESH-M system. It's a very robust and convenient instrument to prepare the lipid nanoparticles. Either you have or have not the payloads. And it has a tunable size feature. You always give you a nice homogeneous solutions with low PDI and high encapsulation efficiency. So the platform is an open platform. It can be upgraded, and the microfluidic chips is transferable by either model. And also, the setup is very simple, easy to use, and the size is compact. And with a touch screen, you do not need to hook up to any external computers. Not mention that the cost is much more efficient, and also either the configuration or the consumables, they're already affordable. So again, thank you today for Victor joining today's webinar. We're happy to have you to share your research experience with us and hope to see you guys next time. Thank you for your attention.
Thank you. Thank you. Thank you, everybody. Yeah.